Hello, and welcome to the Drug Discovery World podcast. My name is Giles, and I'm here to take you through another topical and insightful article from Drug Discovery World. Today's episode is taken from a recent article in our winter 2016-17 issue, titled A Strategic Vision for 21st Century Drug Discovery, Part 2. The article was written by Dr Wayne Bowen, Dr Giles Sanders, Dr Andreas Werdich, Dr Brian Warrington, and Dr Elizabeth Farrant. With so many contributors to this article, I'll be talking through their backgrounds at the end of the episode rather than at the beginning. So now on to A Strategic Vision for 21st Century Drug Discovery, Part 2. Where are we now? Large pharma is in the odd position of being about the only industry that is invested in high-throughput automation without gaining the expected increase in output and lower costs that became expressed as Moore's Law. That is not to say that performance output did not increase for individual tasks. In fact, the productivity measured as the number of discovery items completed, compounds screened, data points plotted, compounds synthesized, hits identified, and leads or candidates delivered increased rapidly on a year-on-year basis. By the measure of the earlier iterative process, which could identify breakthrough drugs by preparing and testing less than 1,000 compounds, under the new regime, a better than 1,000-fold drug output might have been expected. In fact, despite a huge hike in the cost of R&D, the rate of output of drugs did not substantially change. The time to market has remained at 12 years, and there was good evidence that their market novelty and exclusivity was often impaired. Some might say that an active stochastic intelligent iterative search was better than unguided compound synthesis and fatalistic screening. However, the response to this deficiency has been mainly to seek further increase HDS and HTC throughputs, seeking a library size where random chemical profligacy will actually enhance corporate productivity, particularly in respect of novelty. As discussed previously, it is unlikely that this position can be reached. Attempts to improve hardware and software for the iterative paradigm have been held back by the absence of resources and the difficulties encountered in trying to conduct exploratory chemistry using array synthesizers designed to carry out chem-informatic-based exploitation of known reaction types. It is somewhat ironic that after more than 2 decades of development of the linear strategy, that the state of an art in lead and candidate identification is the iterative learning process supporting FBLD. It is now surely time that the shaman's art received the benefit of modern tools fitted to that purpose, so that in-house knowledge, experience and practices of corporate discovery teams can be built into products to generate durable exclusivity in the market based around those important medical needs inaccessible by screening. In fact, with corporate medicinal chemistry know-how as the competitive arena, the development of appropriate and better tools for iterative discovery should be, like the test tube, a mutually beneficial pre-competitive activity. Back to the future. Rational design and iterative discovery in a modern world. The 4 tasks of an iterative paradigm of design, make, test, analyze are necessarily rate-limited by the make step, because the target-specific molecule, or small set of molecules, predicted by unfettered application of structure-activity relationships, as updated in the previous cycle, is extremely likely to be novel. Therefore, a route for its synthesis will need to be investigated. This is itself an iterative design, test, analyze subloop. It is the longer duration of this process, however, that allows time for detailed, multi-parameter biological analysis of molecules and the modelling of putatively improved molecules. In addition, several different stochastic chemistry threads directed to the same target could be pursued simultaneously using the same assay complement for test. Due to the novelty of each molecule in the make phase, a wide range of synthetic methods and technologies need to be available to the chemist. Previous attempts to automate chemistry have focused on trying to force-fit chemical endeavours into a limited range of chemistries carried out using inappropriate high-throughput synthesis systems. This has limited the available chemistry space to those reactions that can be performed in this manner. To fully explore chemistry space, A chemist must use the full range of reactions combined with a full range of chemical technologies, including batch, flow, microwave, photochemical, and extrachemical reactors, along with the relevant analytical and downstream processing and isolation technologies. In the past, this has been a challenge due to the lack of integration and software flexibility. Advances in the Internet of Things and anthropomorphic automation, however, may provide new opportunities in this area. Another challenge when accessing entirely novel chemistry space is reactant supply. Often, reactants for novel molecules are in themselves novel and have to be custom synthesized or are available only in small quantities. In the discovery phase, material-sparing technologies are key to fast progress. However, multi-well HTC synthesizers based on liquid handling robots can be useful in relaying the production of a congeneric range intermediates prepared via a single reaction type. There have been attempts to reduce the material requirement and time needed for exploratory chemistry. The shortened reaction and purification times, analyte frugality, and low-loss transfers possible in nano and microflow systems were an attractive basis for exploration of fast-cycling lead and synthesis discovery systems. In these, it has been possible to adopt the many analyte-sparing protein-based or cell-based assays formatted for HTS. The fusion of solvent-based colliding or printed droplets may also be a new technology worth developing for root scouting. Clearly, larger working scales and equipment can become relevant if the supply chain for intermediates can support them without limiting rate, whatever hardware path is taken. Key accessories requiring development are equipment with robust operating and recovery procedures, a self-educating reactions prediction and methods database, and the operational platform, sensors, and actuators that are essential to a goal-seeking automated system. From a chemistry optimization standpoint, embracing knowledge from every reaction is critical, e.g. the use of knowledge of what also is produced by side reactions to enable improved root scouting for any future synthesis. There is a need to integrate synthesis prediction and machine learning, and this should be a focus for future work, as should integration of the many new information sources that have grown with the internet that could inform the piloting of an automated closed-loop methodology, or be presented for the consideration of a human operator. Machine learning can also have an impact on post-reaction processing and analysis of reaction products. The model: nuclei or electrons. If the full potential of the iterative model is to be achieved, predictive modelling techniques are required that can at extremes either propose a scaffold hop or display the full electronic, steric, and conformational effects arising from the adjustment of just one H atom. Molecular recognition at the site of biological interaction is a complex process, involving both thermodynamic and kinetic effects. Modelling these complex effects requires a series of trade-off decisions to be made in order to enable calculations to run in reasonable time. This is especially true when dealing with large numbers of molecules, or potential molecules, highly flexible molecules, e.g. short peptides, or when modelling complexes of drug molecule and biological target. Molecular recognition and associated biological activity is related to both the 3D shape of the molecule and the electronic structure of the molecule. In a commonly used simplification, the properties of the ligand are dealt with in isolation from any inductive or cooperative effects due to binding. Suitable computational systems then need to be able to model and compare the 3D shape and electronic properties of whole molecules, and, in the context of an iterative closed-loop system, also need to be able to run calculations in reasonable time. This limits choice to modelling systems that can view a molecule holistically as a 3D electronic pharmacophore, and at the same time retain essential structural size and shape, yet deal with the computational challenge with sufficient approximation that the calculation time is tractable. A number of computational chemistry approaches are available to meet this challenge, each with its own set of compromises. As an example, the software from Cresset has been designed explicitly to deal with the challenges of creating a population of reasonable molecular conformations, and then efficiently assessing and comparing these using both shape and electronic properties. A consequence of this is that the Cresset tools are 2D structure agnostic, and are therefore extremely well suited to the task of moving between different molecular scaffolds while retaining features essential for molecular recognition and biological activity. The 3D holistic pharmacophore is very similar across the diverse structures and can successfully hop from one chemotype to another while retaining biological effect. Several of the modelling software suppliers provide tools that can be used to achieve this result, for example Schrödinger, CCG, and OpenEye. But none of these is integrated nor accurate enough, because each relies on crude atom-centered charges which cannot describe experimentally observed electronic distribution. The Cresset technology is founded on the principle of 3D molecular holisticism and has experience in this area. It would now also be possible to draw on ontological and collected data pools to influence the direction of iteration, such as PubChem, ChemSpider, DrugBank, and UniProt. Testing. Effective use of time and technologies. Molecular assays underpin hit discovery in a linear paradigm due to their simplicity and cost-effectiveness, providing primarily insight on target engagement. In the iterative closed-loop model, however, there is an opportunity to leverage information-rich, disease-relevant cellular approaches due to the low number of molecules requiring profiling, typically less than 1,000, and their phased synthesis. For example, it enables testing on primary cells derived from human tissue biopsy at the primary stage despite limiting numbers. Furthermore, these biomaterials can be transformed using advanced gene editing tools into isogenic cell lines to enable the study of both disease and control responses to a new molecule against an otherwise identical genetic background. Emerging 3D cell technologies, such as organoids and organ-on-chips, can also be exploited to mimic human physiology on a microscale. Finally, the use of animal models for phenotype-based screening in the initial stages could circumvent many of the failures associated with an exclusively target-centered drug discovery strategy. The zebrafish is a vertebrate whose organs are remarkably similar to their human counterparts at the anatomical, physiological, and molecular levels. Recently, gene editing techniques have been employed in combination with fluorescence-based reporter systems to create a new generation of genetic and chemical screens. These techniques have demonstrated great potential in uncovering novel modulators of human disease pathologies. The future. In 2006, a review of FDA-approved drugs showed that of 324 targeted proteins, 266, 82%, were human genome-derived proteins, and of these, 48% fell into three classes: GPCRs, nuclear receptors, and ion channels, and that 56% of drugs under investigation were for targets in these fields. The remaining 52% of targets and 44% of drugs were split at levels of less than 5% over 120 domain families or singletons. A recent review in 2017 showed that the number of recorded human genome-derived targets had increased to 667, and 34% fall into the same three classes: GPCRs, nuclear receptors, and ion channels, and 67% of the 700 molecules in investigation are for these fields. However, the remaining 66% of genome-derived targets now contains significant, more than 10%, targeting of cancer-related proteins, for example kinases and proteases. But the number of compounds in the study is low, presumably because they are promiscuous. In both cases, the targets not accounted as human genome-derived were for pathogenic targets. Overall, these data present a picture of a pharmaceutical industry focused almost exclusively on human genome-derived proteins as targets, particularly those dubbed on historical grounds to be privileged protein families, and modulated by structures described, based on long experience, as being somehow permissive. As these targets are likely to be downstream of genetic causes, this pushes discovery towards the treatment of symptoms rather than cures. In addition, the approach excludes non-protein human targets and mitochondrial DNA and its gene products. As the sole energy source of human cells, mitochondrial dysfunction could be, and indeed seems to be, the cause of many of today's sporadically occurring incurables, such as various dementias and metabolic diseases. To use high-throughput screening of huge chemical libraries as the primary filter to find leads and drugs confines drug discovery to those target classes privileged to have simple microplate-based fast assay procedures. It is no surprise permissive structures that modulate this privileged class achieve the classification of drug-like and thus define library selection criteria. It is unfortunate that the practices and influences of large pharma for economic reasons defines the availability of hardware and software tools of medicinal chemistry for everybody, as this impedes the progress of academic and small biotech companies pursuing novel cures and treatments that cannot be launched by random screening and require a more rational, iterative approach. Our own review brings us to the opinion that its preference for screening-based discovery may be self-harming. The performance of an earlier generation of researchers, typified by such as Sir James Black, who actively managed the course of discovery to its goal by use of in-loop feedback from a contemporaneous panel of assays, has proved to be no worse than the linear paradigm in terms of pharma's drug output. In fact, based on its ability to support compound lean exploration of large areas of chemical space to find novel drug chemotypes, some would say it was a better tool for lead discovery. The recent return of the iterative paradigm as the enabler for FBLD probably seals that view. A strategic vision. Knowledge is power is a popular misquote of Francis Bacon, recognizing that information-led approaches can achieve great results. In the modern age, life science is populated by a plethora of omics, with each spawning results into the so-called big data pool. Given there is no apparent end to knowledge, its generation can become self-fulfilling, with a consequential loss of ability to comprehend its meaning. Indeed, the great challenge in a modern world of big data is its conversion to useful and actionable data. In the context of drug discovery, it is therefore imperative to take advantage of modern instrumentation and data processing in a manner that provides a route to undertaking discovery whereby the information is pertinent, valuable, and grows useful, actionable knowledge. A strategic vision is proposed based on a return to iterative discovery enabled by use of modern technology, such as microfluidics and image processing, enabling faster synthesis, route finding, analysis of complex bioassays and decision-making, and harnessing information at all points from multiple cycles of make and test to enable better decisions and more effective future iterations. In addition, create potential starting points for new discovery areas by complete and valuable big data to generate useful leads and drugs. Central to its successful implementation are 5 guiding principles. 1. Iterative cycles of discovery. Make and consider cycles of discovery. 2. Information at every point. Given the interdependence of each process, there must be full integration of thinking between biology and chemistry, be this virtual or real. From a chemistry standpoint, know the failures and embrace knowledge of every reaction. For example, use knowledge of what also is produced by side reactions to enable improved route scouting for any future syntheses. In turn, apply complex assays and use all the information, positive, negative, and unexpected. Chemical and biological space should not be sacrificed for automation. 3. Pragmatism. During implementation, do not attempt to link every piece of equipment unless there is a real advantage. Engineer new solutions where the benefit of doing so is clear. Consider carefully the real requirements at each step, for example, the need to purify a new molecule. If the synthesis is reproducible, for example, as typically enabled by flow regimes, then remake and purify only when required. Understand that chemistry is typically the rate-determining step. Use this as an opportunity to explore more complex biology. Moreover, moving from millions of compounds to typically less than 1,000 enables ready adoption of use of patient-derived cells and simple animal models at a statistically relevant number throughout the discovery process. 4. Blur boundaries of thinking. Continually challenge the known. Integrate target, system, and phenotype profiling of new molecules. Think beyond chemical structure to the pharmacophores. Be willing to jump structures entirely. Electrons, not nuclei, run chemistry. 5. Embrace suitable tools. For make, anything goes. Flow chemistry, library expansion, fragment screening. Chemistry is complex, so do not oversimplify to enable simpler instrumentation at the cost of chemical space. Selected HTS tools can help screen new molecules early in complex organisms such as zebrafish as relevant, scalable in vivo systems to complement mammalian studies. The authors are the first to admit the vision is not necessarily new. In fact, it is close to the drug discovery approaches of the past. However, it engages new data analysis, better screening tools, and rapid synthetic approaches, where pragmatic, to enable faster cycles of iteration and a holistic growth of pertinent knowledge. Our vision is that a pharma-supported campaign to provide a 20 years overdue upgrade of the hardware and software would provide a far superior engine for lead discovery efforts, not only by increasing drug output, but also as a route to distinctive products with durable exclusivity reflecting corporate talent. This was part 2 of 2 episodes, titled A Strategic Vision for 21st Century Drug Discovery. The articles written by Dr Wayne Bowen, Dr Giles Sanders, Dr Andreas Werdich, Dr Brian Warrington, and Dr Elizabeth Farrant. Dr Wayne Bowen is a life science consultant at the Technology Partnership, TTP. His PhD on receptor biochemistry was received from the University of Glasgow. He subsequently worked in the neuroscience department at SmithKline Beecham, developing novel therapeutics for the treatment of depression, Parkinson's disease, and stroke. In 1996, Dr Bowen co-founded Pharmagene plc and coordinated biochemical research on human tissue. Prior to joining TTP, he spent 13 years as Chief Scientific Officer at TTP Labtech. Dr Giles Sanders is responsible for business development and project management in the diagnostics and life sciences market sectors at the Technology Partnership. He has a degree in chemistry and Doctor of Philosophy in reactions at the solid-liquid interface under hydrodynamic conditions from Oxford University. Prior to joining TTP, Dr Sanders worked for a biotechnology startup company where he was responsible for its microfluidics and microfabrication developments. Dr Andreas Werdich is a life science consultant at the Technology Partnership. He received his PhD in biological physics from Vanderbilt University in 2006 and completed postgraduate training in genetics, molecular biology, and cardiac electrophysiology at Harvard Medical School and Case Western Reserve University. More recently, Dr Werdich was assistant professor in cardiovascular research at Brigham and Women's Hospital, Harvard Medical School. Dr Brian Warrington joined SmithKline & French in 1965 as a medicinal chemist and has contributed to a large number of therapeutic targets while developing an increasing interest in the methodology of early drug discovery. He retired from his position as Vice President of Technology and Development for GlaxoSmithKline Discovery Research in 2005 and now pursues the same interests as an independent consultant. Dr Warrington holds a PhD from the University of London, is Fellow of the Royal Society of Chemistry, a chartered chemist, and was awarded the Royal Society of Chemistry Millennium Medal for leadership in the area of microfluidic synthesis and screening. And finally, Dr Elizabeth Farrant. Dr Farrant is Chief Executive Officer at New Path Molecular Research. She has a degree in chemistry from Salford University and a PhD in natural product synthesis from Reading University. Dr Farrant has pioneered the application of microfluidics to chemistry, protein crystallization, separation science, and cell-based assays during employment at GlaxoSmithKline, Pfizer, and Cyclofluidic. If you've enjoyed this episode and want more from Drug Discovery World, then you can subscribe to the journal and get the latest issues delivered to you free. Just head to our website, ddw-online.com, and there's where you can subscribe, as well as read further articles and view original PDFs. If you've enjoyed the podcast and found it useful, then please leave us a review and subscribe. You can also follow us on LinkedIn and Twitter for latest articles and news from Drug Discovery World. Thanks for listening, and we hope to see you in the next episode.